Medical entomology is a branch of entomology which deals with arthropods which affect the health and well-being of man and vertebrate animals. In other words, medical entomology is the medical science directly concerned with vectors that affect human and animal health. In tropical countries, the largest groups of illness are probably insect-borne. It is therefore important to know the habits of insect vectors and how they transmit diseases. It is difficult to implement control measures of insects without some knowledge of entomology and specifically medial tropical countries. The largest group of illness are probably insect-borne. It is therefore important to know the habitats and habits of insect vectors and how they transmit diseases. It is difficult to implement control measures of insects without some knowledge of entomology and specifically medical entomology. To this effect, this course is concerned with the study of arthropods, especially of insects that are of public health importance. Mechanical disease transmission. Disease agents are carried from one host to another by arthropods, simply mechanically carried by the body parts, example wings, hairs, feces, vomitus, etc. And this type of disease transmission to change takes place in the number, form or developmental stages of the organism but simply deposited in the body, food or drink of the host. Mechanical. Biological disease transmission. The agent will exhibit changes in form and number of developmental stages in the arthropod before entry into the host. This include hereditary, transovarian and translycidal transmissions, propagative, cyclodevelopmental and cyclopropagative methods. Transovarian, transstadial transmission. It is a type of disease transmission whereas the causative agent is transmitted to the immature stage, usually to the egg from the adult insect, other arthropods which carry disease pathogens. It becomes ineffective or can transmit the disease to man and other animals. Ticks and sandflies were very good examples of arthropods that exhibit hereditary disease transmission. Then propagative. In propagative type of disease transmission, only the number of pathogens increases and the developmental stages remain constant. The disease, plague and typhus are good example of propagative type of disease transmission. Then cyclodevelopmental. In this type of disease transmission, only the developmental stage form of the disease pathogen is changed, small to big, immature to mature stage, etc. Where the number of the pathogenic organisms remain constant. Example, filariasis, cyclodevelopmental. Then cyclopropagative. This type of disease transmission is a combination of both propagative and cyclodevelopmental, whereby the disease pathogen undertakes a change both in number and developmental form stages. Example, malaria. Life cycle. Cyclical infection of human and female Anopheles mosquitoes. In humans, the parasites grow and multiply first in the liver cells and then in the red cells of the blood. In the blood, successive broods of parasites grow inside the red cell and destroy them, releasing daughter parasites, merozoites, that continue the life cycle by invading other red cells. When certain forms of blood stage parasites like gametocytes that occur in male and female forms are ingested during blood feeding by a female Anopheles mosquito, they meet in the gut of the mosquito and begin a cycle of growth and multiplication in the mosquito. After 10 to 18 days, a form of parasite called a sporozoite migrate to the mosquito's salivary glands. When Anopheles mosquito takes a blood meal on another human, anticoagulant saliva is injected together with the sporozoites which migrate to the liver, thereby beginning a new cycle. Thus, the infected mosquito carries a disease from one human to another acting as a vector, while infected humans transmit the parasite to the mosquito. During a blood meal, a malaria-infected female Anopheles mosquito inoculates sporozoites into the human host. Sporozoites infect liver cells and mature into schizones which rupture and release merozoites. After the initial replication in the liver, exoerythrocytic schizogony, the parasites undergo asexual multiplication in the erythrocytes. Merozoites infect red blood cells. The ringer stage trophozoites mature into schizones which rupture, releasing merozoites. blood stage parasites are responsible for the clinical manifestation of the disease. The gametocytes, male microgametocyte and male macrogametocytes, are ingested by an Anopheles mosquito during a blood meal. The parasite's multiplication in the mosquito is known as sporogonic cycle. While in the mosquito's stomach, the microorganism penetrates the macrogametes, generating zygotes. The zygotes in turn become motile and elongated ookinetics, which invert the midgut wall of the mosquito where they develop into oocytes. The oocytes grow, rupture. Merozoites infect red blood cells. The ringer stage trophozoites mature into schizones, which rupture, releasing merozoites. Some parasites differentiate into gametocytes. Blood stage parasites are responsible for clinical manifestation of the disease. In gametocytes, male and female are ingested by the Anopheles mosquito during a blood meal. The parasite multiplication in the mosquito is known as sporogonic cycle. While in the mosquito's stomach, the microgamete penetrated the microgamete generating zygote. The zygote in turn become motile and elongated ookinesis and invade the midgut wall of the mosquito where they develop into oocytes. The oocysts grow, rupture and release sporozoites which make their way to the mosquito's salivary glands. Inoculation of the sporozoites into a new human host perpetuates the malaria life cycle. Ecology of the malaria. Where does malaria transmission occur? For malaria transmission to occur, conditions must be such that all three components of the malaria life cycle are present. Anopheles mosquitoes which are able to feed on humans and in which the parasites can complete the intermediate host half of their life cycle. Humans who can be bitten by Anopheles mosquitoes, as in whom the parasites can complete the vertebrate host half of their life cycle. Malaria parasites. Climate. Climate is a key determinant of both the geographic distribution and the seasonality of malaria. Without sufficient rainfall, mosquitoes cannot survive, and if not sufficiently warm, parasites cannot survive in the mosquito. Protective effect of sickle cell trait against malaria. The sickle cell gene is caused by a single amino acid mutation, align instead of glutamate at the 6th position in the beta chain of the hemoglobin gene. Inheritance of this mutated gene from both the parents leads to sickle cell disease, and people with this disease have shorter life expectancy. On the contrary, individuals who are carriers of the sickle cell disease with one sickle gene and one normal hemoglobin gene, also known as sickle gene cell traits, have some protective advantage against malaria. As a result, the frequencies of sickle cell carriers are high in malaria endemic areas.